Hello, and thank you for joining us for today's webinar hosted by ISEH. My name is Catherine King, and I'm an assistant professor in pediatric infectious disease at Baylor College of Medicine in Houston, Texas. My lab is really interested in the effects of infections and inflammation on hematopoietic stem cells, so this topic is close to my heart, and I'm pleased to serve as your moderator today. Before we get started with today's webinar, I would like to remind everyone that the ISEH annual meeting will be held this coming August in San Diego. The new pre-meeting ISEH workshop is exclusively for graduate students and postdoctoral fellows, and it will be offered this year on the morning of August 25th. This trainee workshop is mentored by experienced ISEH faculty and will include career development sessions, a poster session, and platform presentations. The best talks will be chosen for oral presentations at the main meeting. More information will soon be available regarding this unique opportunity for trainees to present their current research and network with individuals who will become their future colleagues. The abstract deadline for the meeting is April the 9th, which is at the end of this week, so please submit your abstract, and we would love to see you all in San Diego. I'm now pleased to announce today's session, which is titled The Role of Inflammatory Signals in Embryonic HSC Development and Adult HSC Function. We have over 50 participants registered for today's webinar, as well as several student groups, and we'll have two excellent speakers who will each speak for about 20 minutes. If you have a question at any time during the presentation, please type your question in the question box or the chat box, and I will present your questions to the speakers at the end of the webinar. You're welcome to enter your questions in the chat box at any time, but the questions will be held until the end of the two speakers. So it is now my privilege to introduce our first presenter for today's session, Dr. Marcus Mance. Professor Mance is the director of the Division of Hematology and the Center for Hemato-Oncology at the University Hospital in Zurich. He has made critical contributions identifying many of the mechanisms and cell types through which hematopoietic stem cells and their niches sense and respond to inflammation and infection. I will now turn the presentation over to Marcus.
Hello, this is Marcus Mance talking from Zurich. I hope you can hear me well. Good morning and good afternoon from wherever you are listening. So let me get this organized. Okay, so my first part in the first 20 minutes is about the role of inflammatory signals in adult HSC function, and I will give you a brief introduction and then talk about inflammatory signals and emergency granulopoiesis, and then about inflammatory signals and HSC turnover and function, and then try to summarize it in the end. I also would like to recommend some reading for reviews, one by Catherine, actually our moderator, and Peggy Godell, and another one by Andreas Trump's group, and also we contributed two reviews to the field, which I think all nicely summarizes what has been published. So let me advance this. Okay, so bone marrow actually constitutes one of the largest organs of the human system, accounting for about 5% of total body weight. So that would be about 3 kilograms in a 70 kilogram adult human. And bone marrow tissues are the most active source of de novo cellular generation in adult organisms, outnumbering the rates of cell production of other tissues of high turnover, such as the intestine. And at any given time, about 90% of the human cells in our organism originate from or reside in the bone marrow. And of course, erythrocytes are way smaller than, for example, muscle cells. Now, bone marrow output can be additionally massively enhanced in situations of need, such as massive blood loss or severe infection. And the hematopoiesis is seen as a hierarchical model with all the effector cells at the bottom here that can act immediately with terminal maturation in case of infection, oxidative burst, phagocytosis, or adaptive immunity, if you think of the immune cells. And then above them, the intermediate progenitors with restricted differentiation potential, but high proliferative capacity that feed into the effector cell pool rapidly, and that progenitor cell pool again then is filled up by cells that are on top of them on the furthest part, the hematopoietic stem cells that can self-renew and then give rise to partially self-renewing immediate offspring that then feed again in the progenitor pool. And of course, within these cellular hierarchy, the long-lived self-renewing cells are targets for acquiring or accumulating relevant genetic events that either might lead to death then or might give them a proliferative advantage over other cells and might lead to hematologic malignancies. So I would like to start with a clinical observation. If there's, for example, a local nasopharyngeal infection by bacteria and it is controlled locally, then we do not see major alterations in the peripheral blood and also in the bone marrow. There's basically no change in hematopoiesis. However, if an infection is not locally controlled and bacteria, for example, get access to the blood flow, then we do see massive neutrophilia and a left shift towards immature neutrophils, and if we look in the bone marrow, we see massive enhanced myelopoiesis. So the system acts to this systemic challenge by a systemic reaction that targets to clear then this infection and get control again. Now, how are these signals transmitted? And we do know that there are pathogen recognition receptors, some of them being the toll-like receptors that act basically as smoke detectors for infection. And there are a variety of such receptors, and one of them is very well known, that is TLR4, that recognizes LPS for substances of gram-negative bacteria and then transmit signals to the cell, and the cell is able to react. Now, toll-like receptors are expressed on mature hematopoietic effector cells, and actually there they are very well studied, but they are also expressed on hematopoietic stem and progenitor cells, and there are various publications that have shown that these receptors are there and actually transmit signals upon ligation, leading to mostly differentiation or migration of these hematopoietic stem and progenitor cells, but also, and that's not as well studied, they are expressed on non-hematopoietic cells and can act there to control infections. So the question we had, and that I would like to really lay out here, is how is the hematopoietic system then detecting the spread of germs, and I will mostly focus on lipopolysaccharides, so TLR4 and LPS. Is it that in peripheral tissues these PAMs are then ligating on mature hematopoietic cells that then start to produce cytokines that act on hematopoietic cells or on non-hematopoietic cells, and they produce some factors as cytokines that act on hematopoietic stem and progenitor cells, or are these in this case LPS acting then in the bone marrow again on more mature hematopoietic cells or on non-hematopoietic cells that produce cytokines, or are these pattern-associated molecular patterns possibly acting directly on the hematopoietic stem and progenitor cells that then in a peregrine fashion might produce some cytokines to lead to differentiation, or is it possible that they act directly on them and then without producing cytokines there might be differentiation? So these were the questions we were looking into, and we were doing this, and I will sum that up as this is published, by selectively getting rid of a downstream signaling and adapter molecule, MyD88, in various tissues, either hematopoietic or non-hematopoietic, so that then we could selectively see which cell type is sensing LPS and leads to this massive enhanced granulopoiesis. And the findings were the following, and summing them up, that endothelial cells express TLR4 and MyD88, and following in vivo LPS or live E. coli infection are the prime source of GCSF as it is depicted here. And mice that lack MyD88 in endothelial cells do not respond with emergency granulopoiesis following in vivo LPS or E. coli stimulation. Thus, the endothelial cells catalyze the detection of systemic infection into demand-adapted granulopoiesis. I think this really shows a sophisticated degree of functional interplay between the vascular, the hematopoietic, and the immune system. And I also think that the endothelial cells are, of course, ideally positioned to mark the threshold of local versus systemic infection, and using endothelial cells basically as safeguards seems a very plausible danger-adapted defense strategy established in evolution. Now, what about then the earlier cells, the hematopoietic stem cells and the very early progenitor cells? To address this, we set up a system where we were able to track actually division by division single hematopoietic stem and progenitor cells, and that was done by simply sorting LSKs, so cells that contain hematopoietic stem cells, and then CFSE labeling them and transferring them into non-conditioned, and that's important, steady-state mice, and then analyze them after different time points, and that's shown here between week 1 and week 21. Again, published data, so I'm not going into the details, but the important part here is that when these cells are transferred and observed for a while, for example here 3 weeks, you can see division by division, the non-divided cells, once divided cells, twice divided cells, 3 times divided cells, up to 5 divisions, and afterwards it gets diluted and it's not possible to dissect the divisions anymore. I think it's important to recognize that this might be a more precise labeling technique compared to BRDU, and also it gives the opportunity to resort the labeled cells division by division and test them for their functionality. And by doing so, we figured out that some HSCs are deeply quiescent, and they do not divide for more than 1/5 of the lifetime of a mouse, and during this time for sure they cannot contribute to hematopoiesis. Possibly their offspring could, but they themselves cannot during that time, and one assumes that this might be a mechanism to protect them from metabolic or toxic events, so basically a safe stem cell harbor. And some HSCs, they divide frequently and contribute to hematopoiesis. Some divide every other week, and there is at any given time a heterogeneous division within the HSC pool. Some of the frequently dividing HSCs become slow dividing again, so if you sort the fast dividing ones and put them in secondary recipients, they might become slow dividing again. So the divisional activity in steady state of the HSCs is not fixed, and if that changes all the time, then the divisional history of all HSCs is likely similar at end of life, and that I think is in line with the data on telomere shortening. So altogether this suggests a dynamic repetition model of HSCs that for some time contribute and then go again into a quiescent state. And I think with regards to the long-term quiescence, this is in a good agreement with other publications, for example from Andreas Trump's group and also from Catherine Moore's group and Hannah Hock's group. Now, we used this model to then test what happens if the mice during this time are either challenged with LPS or are infected with live bacteria. And what we observed is that actually when the mice are challenged, here shown for LPS, then the zero divided cell fraction is basically vanishing. So either they die or they go into cell division. And indeed, if we then sort out this population and this population under PBS stimulation, then we do see that the active stem cell fraction as determined by secondary and then tertiary readouts is going into division. So LPS application induces cycling of bona fide functional HSCs. We also tested this in another way by BRDU labeling and then LPS application and subsequent analysis, and there we can observe the very same thing as with the CFSE assay. And interestingly, if we give LPS immediately before application of a chemotherapeutic 5FU, we do see that this causes enhanced toxicity, and we also looked whether this is indeed due to hematopoietic deficiency, and that is the case. So these cells are driven into cycle, and then they are more prone to react to chemotherapy, and the mice die of hematopoietic deficiency. So what one can say from these analyses is that during a gram-negative challenge, for example sepsis, these cells that normally vary between quiescence and activity, they all are recruited to divide and contribute to hematopoiesis during this severe challenge, and then they return to this steady-state dynamic repetition model. Now the question, of course, is how does that happen? Is this a consequence of direct stimulation of stem and progenitor cells by pathogen components? And there is ample literature that says that at least these receptors are expressed and the cells can react. Or is that happening through cytokines that are produced by other types of cells and then act on the stem and progenitor cells? And again, there's quite some literature on that this might be a possibility, and of course it might not be exclusive, but both mechanisms might be active. So let's look first at the cytokine-mediated HSPC activation. We asked the question which of the clinically applied and upon infection and demand increased cytokines or chemokines drive HSC into self-renewing divisions, and we used GCSF, CXCR4 antagonist, c-MIPL agonist, so thrombopoietin-like structures, and FLT3 agonists, so FLT3 ligand. And we checked whether the receptors for those cytokines, respective chemokine, are expressed on the cells, and you can see here the stem cells and more mature. Is that c-MIPL, the receptor for thrombopoietin, is highly expressed on those highly selected or enriched HSC populations, while GCSF receptor is somewhat low expressed, and also CXCR4, at least by RT-PCR, is low expressed, and in the stem cells there's almost no signal for FLT3 recognized. So we again did the CFSE labeling in and transfer in the mice, then gave them a week rest, and then stimulated with rather high doses of those receptor agonists for 1 week, and then observed another week, and then did the analysis and sorted based on the divisional rate of those CFSE labeled cells, and put them then in lethally irradiated mice and had the readout for hematopoiesis, and then put them in secondary recipients to test the stem cell activity. And what you can see here is a summary: PBS, GCSF, CXCR4 antagonist, c-MIPL agonist, and FLT3 ligand stimulated cells. You can see the distribution in the PBS stimulated, where there is very few in the 0 or 1 divided fraction, and the same is true for GCSF, the same is true for the CXCR4 antagonist, and the same is true for FLT3 ligand. However, in the c-MIPL agonist stimulated mice, there's no cells recognized anymore in the 0 to 1 divided fraction, and this is depicted here also in another way, summarized from more mice. Now, when we sort those cells in the PBS, GCSF or c-MIPL agonist treated mice, of course we only can sort 0 to 1 divided cells in the PBS and GCS stimulated animals, but not in the c-MIPL agonist stimulated animals, because there's no cells in this fraction. And then we can sort the 2 to 4 times divided fractions in all 3 types of treatment. And what you can see here is that, as expected with the PBS and the GCSF stimulated animals, you do see engraftment in secondary recipients from a lot cells in limiting dilutions that also are engrafted in tertiary recipients, while in the C-MIPL agonist stimulated animals there's no cells available that could serve as a readout here, but they do engraft from the 2 to 4 divided cells, while from the PBS and from the GCSF stimulated animals they don't engraft, and this is also true for the tertiary engraftments. When we look at type of engraftment, we do not find a big difference in terms of the differentiation to myeloid T or B cells, also not to the engraftment levels. And if we quantify it by limiting dilution, then we do see a 3 to 4-fold increase of functional long-term HSCs in the C-MIPL stimulated animals. So in summary, in vivo supra-steady state TPO receptor stimulation, but not GCSF receptor, as well as CXCR4 antagonism or FLT3 ligand stimulation, induces HSCs to exit from dormancy into cell cycle, and TPO receptor stimulation expands the frequency of long-term repopulating HSCs in vivo. Now, what about stimulation then of TLR4 in these HSPCs? What you can see here is TLR4 MD2 expression on HSCs and the subsequent progenitors, and this is also recapitulated here by RT-PCR. And to then see what would be the consequences of stimulation, we co-transplanted wild-type and TLR4 knockout whole bone marrow or LSK into wild-type or TLR4 knockout F1 animals, and then could read out in the granulocyte gate the contribution of the wild-type or the TLR4 knockouts. And what you can observe here is rather complicated, but when these chimeras have reached steady state after a couple of months and then are BRDU labeled and then stimulated with LPS or PBS as a control and then analyzed, one can see that the TLR4 competent HSCs are dividing, and the division is stronger than in the TLR4 deficient HSCs. Actually, the delta marks the difference between the wild type and the TLR4 deficient HSCs, while this delta here reflects the action of the cytokines that are produced in this context by the hematopoietic cells of the wild-type chimeric mice, because the background is negative. When we then look what happens actually to those cells, we again by CFSE dilution see that the cells that are TLR4 competent are starting to proliferate, and they are skewed towards myeloid differentiation. So the question then is actually the microbiome in the steady state also contributing to stimulation of early hematopoiesis? And it is known that germ-free mice have a decreased total white blood cell count and neutrophil counts. They also have some other alterations that are marked here. And when we then looked at granulocytes and monocytes in germ-free mice versus regular pathogen-free mice, one can see that the amount of granulocytes is significantly higher, as well as monocytes, and this is also true for LSK, for GMP, but not for CLP. And if you take out bone marrow cells from those animals and test them in colony-forming assays, then the frequency of colony-forming cells is significantly smaller in the germ-free mice versus the SPF mice. And the same is actually true when mice are treated with broad-spectrum antibiotics for over 4 weeks. So there also the mature myeloid cells get fewer, and also the numbers of LSKs are fewer, and the frequency of colony-forming cells in the bone marrow also is decreased. Now, how does that work? When we take serum from regular mice and inactivate it with heat, then in the germ-free mice you can induce peripheral blood counts that resemble that of healthy animals, and when we then look in mice that do not have MyD88 or TCAM, there is also TRIF, so the other signaling molecule downstream of TLR4, then it's clear that the transfer of serum does not rescue the phenotype. So again, this is acting through MyD88 and or TCAM via TLR4, most likely in this setting. Now, to look a little bit closer into the question of how is the downstream signaling of TLR4 in stem and progenitor cells, we simply stimulated TLR4, MyD88 and TRIF knockout mice and compared them with wild-type mice, and what can be seen is that in LKS, the activation as depicted by upregulation of SCAR1 depends on TRIF, but not on MyD88 in these cells that are defined by an LKS phenotype, and that's also quantified here. So we then thought to take again chimeric mice with wild-type and TLR4 knockout, hematopoiesis 1-to-1 in either wild-type or TLR4 knockout recipients, and chronically stimulate, or over a longer time stimulate with LPS and see what that would do to hematopoiesis. And to make all the story here quite short is that one can see that the HSPCs and HSCs with a TLR4 deficiency in the end win and gets dominant over the wild-type cells, and in the secondary transplants this is seen even more prominent. And when we then take MyD88 or TRIF knockout bone marrow or sorted LKS and get these into chimeric mice and do the stimulation, again we do see that the TRIF knockout hematopoiesis wins over the wild-type hematopoiesis after the stimulation, while this is not the case for the MyD88 knockout, again indicating that the signaling goes through TRIF but not MyD88 in the early hematopoietic progenitor cells. So can this be modified? We tested small molecules that inhibit the downstream signaling of TLR4, particularly scavengers for reactive oxygen species or inhibitors of P38, and again stimulated them in combination with LPS and these inhibitors animals repetitively, and what can be seen is that the pharmacological inhibition of ROS and P38 activation then prevents this LPS TLR4 TRIF induced HSC dysfunction, because then the hematopoiesis is rescued and the knockout does not gain dominance over the wild-type hematopoiesis. So to sum also this up, I think the data shows that direct short-term TLR4 activation contributes to HSC cycling and myeloid differentiation. So that is a beneficial effect on the short term to cope with a challenge, while then when there is sustained TLR4 activation, this contributes to HSC impairment and dysfunction, though these are detrimental effects. And the data I haven't shown here is that actually this chronic stimulation in the end induces genotoxic stress, and again this can be rescued by those inhibitors that I indicated. So how can we put this into context with aging and possibly chronic inflammation. Some people talk not about aging, but about inflammaging. There have been some, I would say, hallmark publications that showed that hematopoietic stem cells continuously acquire single nucleotide variants while individual age. This was published by Welch in Cell in 2012, and then 2 years ago, almost 3 publications showing that in large non-hematopoietic disease populations, oligoclonal hematopoiesis is getting a frequent event with aging. Actually, it was shown that in the population over 70 years, about 10% do have clones that are detected in a threshold that is higher than 5%. So oligoclonal hematopoiesis without causing primarily a hematologic malignancy is present in about 10% of the population over the 70 years old. Also, we do know that inflammation increases with age, here measured by C-reactive protein, and we do know that AML and MDS incidence increases with age. So this is, of course, purely correlative. However, we also know from population studies from Sweden, from the Swedish Cancer Registry, that has a coverage that is over 98%, that a history of infectious disease or a history of autoimmune disease increases the likelihood to get a clonal malignant hematopoietic disease. So again, correlative data, and when we take all this together, one might speculate, but of course that is not proven, that in a chronic repetitive inflammation or infection, this enhanced cycling of hematopoietic stem cells makes it more likely that genetic hits occur. And when they occur, it might be also more likely that in an inflammatory environment, these cells are rescued and actually are acquiring additional events, and then in the end might cause full-blown clonal hematopoietic disease. So in summary, I'm coming to the end. Adult steady-state hematopoiesis is in part driven by microbiota-released conserved patterns. Adult HSCs and HSPCs are able to react to infection and inflammation either by direct pathogen pattern recognition or then by inflammatory cytokine-induced responses, or a combination of it, most likely. And sustained inflammatory activation leads to competitive disadvantage and might be causing, in the end, HSC loss or even HSC transformation. And with this, I would like to particularly acknowledge all the people involved in the work I just presented, and thanks for listening.
Thank you very much, Marcus, for that excellent talk and a great review of a lot of literature. It's exciting stuff. I'd like to thank the audience for entering some questions, and I'll remind you that we'll have time for questions for Marcus at the end of the webinar. So now I have the pleasure of introducing our next speaker, who is Trista North. Trista is an assistant professor in the Department of Pathology at Harvard Medical School and the Beth Israel Deaconess Medical Center. Her lab has discovered the mechanisms through which interferon signaling regulates HSC emergence in the embryo and how prostaglandin E2 regulates HSC function. So I will now turn the presentation over to Trista. Right now, I invite the audience to write in with their questions if you have any for Dr. Mance. And I guess I'll start right now, if you don't mind. I was really interested in a lot of the things you were talking about. One of the questions that I had, Marcus, was in terms of the LPS signaling to HSPCs, do you have any sense of how long those signals last, and if there are regulators of that response, or is it simply the removal of LPS signal that would then allow the progenitors to go back to a quiescent state?
Yes, so I don't have data about negative regulation. So actually, the short answer is I don't know.
Mm-hmm. Yeah.
The situation is quite complicated, as whatever you do over a longer time stimulation, you always have, of course, concurrent signals that play as cytokines, even if you are in a complete receptor-free environment. As soon as those cells start to differentiate, they themselves might become cytokine producers, so that's a little bit complicated.
Yes, right. Yeah. I think that is a complication in a lot of this research, is that the hematopoietic cells that are produced in the process then feed back and have secondary effects.
Yes, so actually I think with the very short-term experiments where you can control for not having more mature cells, let's say as the MPP, at least you can exclude to have some more mature, well-described cytokine producers. But with the data, for example, from Baltimore's lab, it also seems clear that progenitors at least are able to produce some of the cytokines themselves and stimulate themselves in an autocrine fashion. How important that is in an overall response, I think, is again difficult to dissect, but when you have competent environment, I'm not sure how important it is, but when you have a deficient environment that is not able to sense pathogens, then, but you have it in the hematopoietic cells, then it might be quite of some importance.
Right. Excellent. So I was interested in the data you showed about the c-MPL agonist. I know that's an agent that's now being used clinically for patients with aplastic anemia. Yes. And I wonder if you want to comment about how your findings give us perspective about what's happening in those patients.
Yes. So first, c-MPL agonist treatment was on the market for chronic immunothrombocytopenia, and there clearly the effects are quite dramatic in terms of increasing platelet counts, and then now it's in clinical trials for aplastic anemia, and also we have treated a couple of patients actually outside of clinical trials and saw some quite remarkable responses. So my understanding would be that indeed we help homeostatic expansion of the remaining HSCs in the setting of aplastic anemia. So usually it's given, of course, in context of immunosuppression. So aplastic anemia is thought the result of is an autoimmune disease and treated with antithymocyte globulin and cyclosporin usually, and those that do not respond or slow responders might now in clinical studies so far receive also c-MPL agonists, and I think there it's critical to expand the very much diminished HSC pool, and that seems to be what the c-MPL agonists do.
Excellent. Okay, I have a question from the audience. They're wondering about LPS, and given that LPS is only expressed or a product of gram-negative bacteria, is there any information or data about gram-positive bacteria and any alternative mechanisms for HSPC activation by gram-positive bacteria?
Yes. We have not tested that. We have only used so far LPS and gram-negative infections. As a clinical reaction is very much the same also with gram-positive bacteria. I would assume that similar mechanisms are in place, but we have not tested it.
Yeah, I think actually Laura Schutpeltz at Washington University has done some work in this area that may be relevant. I guess we'll see that there's TLR2 expression on HSCs following GCSF treatment. So that will be interesting to see how relevant that is.
Yeah. So the receptor seems to be there, also that we have looked for, but we haven't done the whole experiment.
Yes, yes. Interesting. Okay, let me check in. Trista, are you with us yet? I think we're still working on our technical issues here, so we can go on with some other questions. So I think you alluded to in the last part about a potential connection to cancer development, and there's quite a broad literature about inflammatory signals being important in the development of MPNs and myelodysplastic syndrome. But I don't know, I'm not very familiar with the literature in terms of other kinds of leukemias.
Yeah.
And I wondered about that.
So there's data for acute myelogenous leukemia. There's population-based data, right? I'm talking about population-based data in individuals that had repetitive infections or have inflammatory autoimmune disease. There is good data for AML, for MDS, and also for myeloproliferative disease, all coming mostly from the Swedish cancer registry, which really is a very good registry because they have almost 100% coverage and have long-term follow-ups. So yeah, that's correlative data, of course. And then there is data, you might also from Starzynovsky's lab, that show that actually in these diseases also inflammatory signaling pathways are on, which again can say, you know, this might be a driver for disease, whether the regular stimulation by inflammation, infection, then causes the disease. Again, I think is not, there's no proof of it thus far. I think we have to see the data whether indeed chronic inflammatory reactions would lead to accumulation of potentially transforming genetic events, and this data I haven't seen yet.
Mm-hmm. Right. Interesting.
But in the end, you know, when you think of other types of cancers that are associated with inflammation, it seems in a way obvious, but it needs to be proven.
Right. Right. I feel like that's something that we've talked about for many years or even decades, but little clear evidence. Yeah.
As the frequency of these diseases is so low, you know, even doubling the incidence, of course, brings it then from 1 to 3 to 100,000 to 2 to 6 in 100,000, and that's difficult to grasp really. I wonder whether the data on clonal hematopoiesis without being a malignancy will be easier to study with that respect.
Yes. Okay. Great. One of the other questions that I have, and I'm sorry, we're still working on getting Trista back in contact, so thanks for everyone's patience. One other question I had was that you've nicely shown that the endothelial cells are very important in signaling to HSCs during an inflammatory response. And I think that's very interesting also in light of the evidence from Paul Frenette's lab that showed that the stem cells seem to move away from the vascular endothelium during neuron responses. So I wonder if there's any connection there, or if you have any thoughts about that.
Yes, so first of all, I have to clarify that for the endothelial production of GCSF that is very pronounced after LPS application or gram-negative infection, I'm not sure whether their production of cytokines in the end has an impact on HSCs themselves, or whether this is all acting on progenitor cells. That might well be the case, especially as we do not see an effect besides mobilization on HSCs from GCSF, at least not if we use it for 1 week at high doses. So we don't see a proliferative effect on real HSCs, but we do see that once the cells are somewhat more differentiated. So the part with the endothelial cells, I have to stress, I didn't relate really to stem cells, but rather to progenitor cells. And to your question, again, we haven't looked at it, but certainly it's a very interesting connection. And of course, endothelial cells might produce a lot of other things that might have an impact on HSC. That has been shown already, right? But we didn't look at it.
Great, thank you so much. I have a couple of questions that I wanted to ask Trista before we go back to another question from the audience for Marcus. So one of the observations that you've made and others also is that even though there seems to be a pretty significant effect of the lack of interferon signaling on HSC development and embryogenesis, that in the adult the number of stem cells seems to be normal, or at least close to normal. And this, I think, also applies to humans, that there are people with interferon gamma receptor deficiencies, for example, and to my knowledge they don't have any baseline hematopoietic defects. So I wonder if you have a comment about that.
I'm sorry, can you repeat that end of it? You cut out in the end.
Oh, I'm sorry. I was just wondering about the observation that even though you've seen a lot of deficit in HSCs in the absence of interferon gamma signaling, that in the adult stage the number of stem cells seems to be normal, or at least close to normal, and that's also true in humans, I think. So is there, what's your thought about that in terms of the difference in between the fetal stage and the adult stage in terms of sheer numbers of stem cells?
Yeah, so I don't know how well you can hear me. I keep hearing the question go in and out, but I think one of the interesting findings that we saw in the embryo in both the zebrafish and the mouse was that these factors didn't seem to be all or nothing factors in the way that Notch or some of the major pathway regulators are. They're really definitely modifiers, and that we see about a 50% reduction in stem cell production. So I think that they're really acting to kind of similar like you would have in an infection, really acting to control the overall output or production of stem cells, but the stem cells themselves seem to be produced in their absence. So it's a bit more of kind of reaching the optimal number of HSCs, and I think that probably speaks to why several different cytokines can work. So when we lose interferon gamma, we get a little bit of dip, but as long as TNF-alpha is around, we'll recover. I should say TNF-alpha also looks like it plays a little bit of a role in multiple places, so that one seems to also impact Notch, which we know is really essential for making the hemogenic endothelial niche, so that one can have a little bit more of a potent effect. But I think in terms of the interplay between the primitive myeloid populations, it seems to be a modifier rather than an essential to the specification itself.
Yeah.
I don't know if that got at your question.
Yes, yes. Okay, so I think think the last question that we'll have before we wrap up is this one from the audience for Marcus. And they're wondering about really what we were sort of talking about at the end in the last question was about tissue inflammation and cancer risk. And for example, patients who have inflammatory bowel syndrome or other types of bowel disorders where they may have a leaky gut and they may have a high frequency of bacterial translocation across the intestine, if that then would make such patients more prone to having blood disorders, including leukemias.
So again, I would refer to population studies that indeed found that the risk for AML and MDS is moderately increased in individuals with ulcerative colitis. I see on the screen irritable bowel syndrome, and I'm not the expert on it, but I understood that irritable bowel syndrome would not be an inflammatory disease that leads to a leaky gut, but is rather another problem. But clearly, inflammatory bowel disease, there is population studies that show this moderately increased risk for AML and MDS.
Yes.
It's not like 20-fold higher, right? It's somewhat 1.5, 2-fold higher. So
yeah.
Again, with the incidence of both inflammatory bowel disease and then hematologic malignancies, this is extremely difficult to pin down. And again, I think if this would hold true, and if this type of inflammation would foster the outgrowth of some pre-leukemic cells, that possibly could be shown then with the higher incidence of clonal diseases. And I think that soon there will be data coming out correlating inflammatory markers with this type of clonal hematopoiesis. I'm not sure whether they will be positive. We have to see.
Yes. Another question, I think, related to that, but for Trista, is that we've talked a lot about how infections and microbiome appear to affect hematopoietic progenitors, but clearly those factors do not exist for the embryo. So can you clarify a little bit more about what induces those inflammatory signals in the sterile embryonic state?
I think in some ways we don't totally know. There are some interesting kind of side notes that showed up over the several different papers that suggested that it was regular kind of growth factor and transcriptional cascades that are initially turning these receptors on the populations that are present. We do also know in one study from Didier Stanier's lab, it was interesting correlations with blood flow. So there may be sort of physical cues that start to make some of these inflammatory signals first initiate, and then I think the cells potentiate each other, and that the primitive myeloid populations may be speaking back to the stem cell population. So it may be very intricately timed to the development of the embryo itself, and that once you have flow and once you have myeloid circulation, that's when you start to get this crosstalk.
Yeah. Excellent. Yeah, so thank you so much. I think those are all the questions. So I want to thank everyone for participating in today's webinar, and I'd like to extend a special thank you to our two presenters for today. Thank you so much for two really great talks. So I'd ask the attendees to please complete the evaluation for today's event that you'll receive by email. We really value your feedback and input to further develop ISEH webinar programming. So thank you again for attending, and hope you have a great day.